Hello class. By this point we've talked a lot about the details of the process of photosynthesis. We've talked about the light-dependent reactions, we've talked about the light-independent reactions, and we've also talked about the factors that influence the rate that photosynthesis happens. Our last piece of or topic of discussion in our photosynthesis unit will be described as photosynthesis alternatives. Photosynthesis happens for most plants very well. It's a pretty efficient process. However, there are certain plants, specifically plants that exist in arid environments, where the photosynthesis process that we've described up to this point doesn't work well for them. We're going to talk today about why it doesn't work well for them and the different adaptations plants have developed in order to cope and be well adapted in these kinds of environments. Alright, so we've seen this diagram before. Up until this point, we've agreed that photosynthesis happens in these cells over here. We call those cells, remember, the mesophyll cells. I'm going to make my pen thicker here in a second. Alright, these are the mesophyll cells. And we know that photosynthesis happens in these mesophyll cells. Both the light-dependent and the light-independent reactions happen here. If we come back to this overview diagram of photosynthesis, we have to remember that in the mesophyll cell, in the chloroplast of that mesophyll cell, we have both parts of photosynthesis taking place. We see the light reactions take place in our thylakoid, in our thylakoid membrane, as well as the lumen. And the products of our light-dependent reactions include ATP and ADPH, and they are sent into the stroma of the chloroplast of those mesophyll cells, of those same mesophyll cells, combined with CO2, and we have our Calvin cycle taking place. Okay, so key here is that in most photosynthesis in most plants, photosynthesis takes place inside one single cell. We call those cells the mesophyll cells. We're going to show you in a second an alternative to this kind of photosynthesis. Before we talk, however, about this alternative kind of photosynthesis, I want to talk a little bit more about a different kind of plant anatomy. Like I said, up until this point, we've talked a lot about mesophyll cells. We've maybe talked a little bit about xylem and phloem. But the structures that I want us to pay close attention to are these structures. Okay. We see that there are two vocabulary words here. One of them is called the stoma. Now don't get this confused with stroma. Stoma is a singular noun, and it's plural. I'm going to write it down for you. You're going to hear it a lot. Is stomata. Okay. So plants have these structures called stomata, or they have a bunch of stoma. Okay, and these stomata are surrounded by cells known as guard cells. Let's take a closer look at the anatomy of these structures as well as their function. But before we go to the next slide, though, keep in mind the orientation of our leaf here. We notice that our mesophyll cells. Oops, yikes. All right, let me use my pen instead. We notice that these mesophyll cells are closer towards the top, or the anterior of the leaf, so that the sun shining on top of the leaf, the light of the sun can be absorbed and photosynthesis can happen. Notice that the stomata, or this particular stoma, and all of the rest of the stoma, they are on the bottom end of the leaf, and this is going to have a lot to do with the function and the anatomy of these stomata. So if we take a closer look, if we look at this picture here, we'll notice that this is actually a microscopic view of some of plant cells, and specifically of stomata. All right, so we see a number of stomata in this picture, and the cells surrounding the stomata are other simple kinds of plant cells, plant epidermis. But what's cool is that you can actually see them under a microscope. Now, if we bring our attention to this diagram here, we'll see that the plant stoma, or this plant stoma, is essentially a hole at the bottom of a leaf. This hole is a really important type of hole at the bottom of a leaf, and the hole itself has two cells. We call those cells guard cells. So the stoma is essentially a hole. It's a hole surrounded by two cells known as guard cells. Now, if we take a look at this diagram here, we'll notice that these guard cells have a cool function. These guard cells, in many ways, just like other guards of doors or of passageways, these guard cells can open and close. They can allow things to enter and leave. What you'll see is stoma can be closed, and stoma can then open. They open when the guard cells become turgid, when they become rigid. Once they rigidify, they open the stomata. Then they can ultimately close back up again. Now, what's the point of this? Of these stomata, or what is their function? What is the function of this hole at the bottom of a leaf, or these series of holes? Well, as this diagram shows you, the point of this whole thing is so that carbon dioxide can enter our leaves. You ask yourself, how does carbon dioxide actually get into the leaf to perform the Calvin cycle? Well, it gets into the leaf via the stomata. Not only that, but the waste products generated. Waste products like specifically oxygen gas. They also oxygen gas leaves the stomata, and water transpiration happens. Evaporation from leaves. Water leaves the leaves. Water leaves the leaves via the stomata. Okay, so we see the function of the stomata when it's open is to intake carbon dioxide and to allow oxygen gas as well as water to leave the leaves. Importantly, when the stomata are closed, as you can imagine, it prevents carbon dioxide from entering the leaves. It also prevents water from leaving, as well as oxygen from leaving the leaves. Now, for about 90% of plants, these stomata, as well as the idea that photosynthesis happens in the mesophyll cells. Both the light reactions and the Calvin cycle happen in the mesophyll cells. This works really well for about 90% of plants. However, plants in arid environments they encounter a specific kind of problem here. In arid environments, when it's really hot and when it's really dry, water leaves the stomata quite rapidly via evaporation. That makes sense. When it's hotter and drier, evaporation quickens because there's not a lot of water in the air. So what plants in arid environments do instead, they close their stomata to conserve water. Otherwise, the plant would wilt and die. However, with the stomata closed, the carbon dioxide that the plant needs for the Calvin cycle can't enter the cell or can't enter the plant. And as a result, the plants in arid environments, when they close their stomata, they don't perform photosynthesis. They can't produce the kinds of sugars and carbohydrates needed for a plant's survival. These plants are very poorly adapted to arid environments. In fact, when plants close their stomata for an extended period of time, a sort of negative side effect occurs. This side effect is called photorespiration. In order to understand photorespiration, we have to quickly review the Calvin cycle of photosynthesis. Remember, in the Calvin cycle, we start with carbon dioxide, and we take this enzyme called ribisco and this 5-carbon sugar called RUBP, or ribulose biphosphate, and we incorporate the carbon dioxide into the RUBP. And ultimately, we generate this molecule known as phosphoglycerate, or phosphoglyceric acid. We call that molecule PGA. And ultimately, PGA is transformed into G3P, and we use G3P to form our 6-carbon sugar like glucose or sucrose or something like that. Okay? So this is what happens when under normal circumstances with the Calvin cycle. The problem arises, of course, when plants close their stomata. When plants close their stomata, the plants don't have carbon dioxide. They don't have enough carbon dioxide. And the complication arises when we recognize the fact that RUBP, ribulose biphosphate, doesn't just bind with carbon dioxide. In fact, it also binds pretty well with oxygen gas. And we know that when the stomata are closed, carbon dioxide can't get in, and the oxygen that's produced can't get out. In fact, we have a high concentration of oxygen. And when that happens, oxygen, instead of carbon dioxide, will bind to ribulose biphosphate. It will bind to RUBP instead of carbon dioxide. And if carbon dioxide isn't binding to RUBP, we're never generating our G3P. We can't generate our carbohydrates. Instead, we go through this process known as photorespiration. And photorespiration ultimately gets rid of the oxygen, and it uses it counterproductively. And it ultimately it results in not making carbohydrates, and the plants won't be able to survive. So ultimately what we can conclude is when plants are in arid environments, when they close their stomata for extended periods of time to conserve water, they're more likely to engage in photorespiration, which is the use of oxygen instead of carbon dioxide, and we don't produce carbohydrates. So plants that engage in normal photosynthesis in arid environments are very poorly adapted to those arid environments. And as a result, certain plants, specifically plants that are native to arid environments, have developed an alternative route to doing the same thing, to accomplishing the same kind of photosynthesis. The normal photosynthesis that we've been describing so far is known as C3 photosynthesis. The reason we call it C3 is because the first product in the Calvin cycle, we take CO2, we take RUBP, and we create PGA. PGA is a 3-carbon sugar, so we call that photosynthesis a C3 process, or a C3 mechanism of photosynthesis. Plants that are well adapted to arid environments perform a different kind of photosynthesis known as C4 photosynthesis. As you can imagine, C4 photosynthesis doesn't produce PGA immediately upon starting the Calvin cycle. Instead, it creates a 4-carbon molecule, so we call it a C4 photosynthesis. In order to understand C4 photosynthesis, the bottom line is this. In C3 photosynthesis, we know that both the light reactions and the Calvin cycle take place in mesophyll cells. But when stomata are closed, we can't perform the Calvin cycle because we don't have enough carbon dioxide. So what C4 plants do is they, during times where they can open the stomata, like early in the day or even at night, plants will open up their stomata, carbon dioxide will come in through the stomata and enter the mesophyll cells just like normal. In C4 plants, however, the CO2 that comes in doesn't immediately begin the Calvin cycle. Instead, what happens is this CO2 is converted into our 4-carbon sugar, or into this 4-carbon molecule. And after it's converted into this 4-carbon molecule, it is transferred out of the mesophyll cells into a new cell. This new cell is called a bundle sheath cell. And it's in this bundle sheath cell that our 4-carbon molecule is reconverted into carbon dioxide and now performs the Calvin cycle. Okay, so instead of performing both the light reactions and the Calvin cycle in the mesophyll cells, in C4 plants, the carbon dioxide is converted into a 4-carbon molecule, transferred into a new cell where it can then create or perform the Calvin cycle. The reason it does this is by converting carbon dioxide into a 4-carbon molecule, we can ultimately create a high concentration of CO2 in these bundle sheath cells. And if we have a high concentration of CO2 in the bundle sheath cells, then normal photosynthesis can take place, and photorespiration will not likely take place at all. Let's take a closer look now at C4 photosynthesis. So, when carbon dioxide comes into the plant via the stomata, that carbon dioxide will enter into the mesophyll cell, and a new enzyme, an enzyme that's not present in in C3 cells, does a job. This enzyme is called PEP carboxylase. PEP carboxylase takes carbon dioxide, our one carbon molecule, and converts it into oxaloacetate, also known as OAA. Oxaloacetate is then ultimately converted into malate. Malate is the 4-carbon molecule that we're talking about, because malate is the molecule that can transmit itself from the mesophyll cell into our bundle sheath cell. So let's take a look at what we're doing here. We're taking carbon dioxide, we're converting it via PEP carboxylase into oxaloacetate, and from oxaloacetate into malate. Malate then is transferred into the bundle sheath cell, where a new enzyme reconverts it back into CO2, and the Calvin cycle can take place just like normal. And once we have this sugar generated, we can send that sugar into a vessel of the leaf, and it can be transmitted into various portions of the leaf. So this is C4 photosynthesis. Instead of immediately performing the Calvin cycle in the mesophyll cell, we convert it into a 4-carbon molecule where we transfer it to the bundle sheath cell, and so that the Calvin cycle can take place without photorespiration taking place. Most flowering plants that you can find in arid environments, in hot, dry environments, most of those plants are C4 plants. They perform C4 photosynthesis instead of C3 photosynthesis. There is a second kind of alternative to photosynthesis that we'll now address as well. This second alternative to photosynthesis is not called C4 photosynthesis, though it's similar to C4 photosynthesis. Instead, it's called CAM photosynthesis. CAM itself stands for, now this is a difficult word to pronounce, crassulation acid metabolism. Crassulation acid metabolism. CAM. In C4 photosynthesis that we just talked about, we know that plants separate the light reactions from the Calvin cycle by separating the actual cells that are performing these jobs. The mesophyll cells are performing the light reactions, and the bundle sheath cells are performing the Calvin cycle. Something similar happens in CAM photosynthesis, except instead of separating the Calvin cycle into a separate cell, what CAM plants do is they separate the Calvin cycle into different times of the day. So what we see here is that during the nighttime, these plants open up their stomata. When the plants open up their stomata at night, the plants can now take in atmospheric CO2. Okay, we can take in CO2 from the stomata at night. A similar conversion happens in CAM plants, where our CO2 is converted into malate and into malic acid. It's converted into a 4-carbon sugar, or a 4-carbon molecule. However, instead of sending this 4-carbon molecule into a different cell, it actually sends that malate into the vacuole of the plant cell. In the vacuole of the plant cell, this malate, or this malic acid, is stored until it can be used during the day. So ultimately, when the stomata are open, our CO2 comes in through the stomata and is converted into malate, our 4-carbon molecule, and is stored in our vacuole. During the daytime, when plants can perform their light reactions, because we've got sunlight now, what happens is plants in the chloroplasts, the chloroplasts are performing their light reactions, the light-dependent reactions, and the products of the chloroplast, the products of the light-dependent reactions, are sent back into the stroma. And as soon as those products are sent into the stroma, the malic acid is converted back into malate, and that malate is converted back into CO2, and the Calvin cycle can take place. So as we can see in CAM photosynthesis, carbon dioxide is taken into the plant at night, stored overnight as a 4-carbon molecule, and is reconverted back into CO2 in the daytime, even though the stomata during the daytime are closed. All right. Here's another diagram of CAM photosynthesis. We see carbon dioxide entering in through the stomata. It is entering into our mesophyll cell, where again PEP carboxylase turns our carbon dioxide into oxaloacetate, and then into malate and malic acid, where it's stored in a vacuole. This is happening, of course, at night when the stomata are open. During the day, the stomata are closed, and our vacuole that has malic acid stored into it converts it back into malate, and then from malate can convert it back into CO2, where the Calvin cycle can now take place. Again, so CAM photosynthesis is separating the Calvin cycle via temporality, via time. So carbon dioxide is taken in at night, and the Calvin cycle takes place during the day. What I wanted to quickly show you in this final slide of our lecture is that plants like, for example, sugarcane, those plants take place in hot, arid environments. Those are C4 plants. Other plants like pineapple plants, many cacti, for example, also are CAM plants. And again, both of these processes, both C4 photosynthesis and CAM photosynthesis, prove to be very well adaptive means of surviving and thriving in arid environments. So by this time, we've learned about 2 important alternatives to C3 photosynthesis.